Hello, welcome back to another, not Pal World, another science stream. Let's go. And today we are going to discuss protein targeting and translocation. Okay? But actually, before that, there is a mini running, not an intro, but a running event that's happening. So we recently hit 12,000 subscribers, right? And then what I thought was like, okay, why not to celebrate? I'm gonna do 1200 jumping jacks. And then right now we finished sets 1, 2, 3, and 4. So that means we are at it's gonna be 120 jumping jacks each time. So times 4, that's 480, right? And right now, I'm about to do set number 5. So let's go. One moment. Let me stand up. Okay, and the biggest proof for that would be the heart rate monitor, okay? Let me just move the mic really quickly. All right, this one works, right? Okay. And let's go. Wait, space, we need space. Okay, go. 1, 2. Oh, my bracelet. Okay. 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. Wait. And 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40. Hello, Paulize. Yes, right now, so I'm not sure if you saw the mini announcement. Wait, what number did I say? 40? Let me just write that really quickly. So, welcome back to the stream, by the way. So, we recently hit 12,000 subscribers, right? And I was like, why not do 1200 jumping jacks to celebrate? But I was like, it would be crazy if I just tried to do them all at once. So, I broke it up into about 10 sets. And I'm doing the 5th set right now. So, yeah. And then I'll continue doing it until it's 120. And then we'll proceed to the protein targeting lesson. Okay? So, what number was I at? 40, okay. And let's go. 41, 42, 43, 44, 45, 46, 47, 48, 49, 50, 51, 52, 53, 54, 55, 56, 57, 58, 59, 60, 61, 62, 63, 64, 65, 66, 67, 68, 69, 70. Calm down. We're all right. So it's 70. Let me just write it. There. Okay. A little bit more and it's gonna be finished. And 71, 72, 73, 74, 75, 76, 77, 78, 79, 80. Wow. Okay, it's 80. Where is the heart? Oh, there you go. I'm gonna put it bigger actually. 80. It's 80. Okay. Just 40 more and it's 120. Finish the 5th set. And 81, 82, 83, 84, 85, 86, 87, 88, 89, 90, 91, 92, 93, 94, 95, 96, 97, 98, 99, hundo. Okay. Almost finished. Wow. At least I think it's getting better. The very first set was like the heart rate reached 163, 168, or 167. Something crazy. I was like, okay, it's working. Alright. 20 more. Let's go. And 131. Wait, nope, wrong. 101, 102, 103, 104, 105, 106, 107, 108, 109, 110, 111, 112, 113, 114, 115, 116, 117, 118, 119, 120. Wow. We did it. Finished. And set 5 is finished. Whoop whoop. Okay. On to science. Protein targeting and translocation. Okay, we know that. Let me breathe first. One moment and readjust the mic. Wow. Okay, so we have the ER. How's the mic, by the way? Is that working? Can I hear myself? Okay, it works. So for protein translocation, we know that the ER is, well, let me just erase this. 1 second. We know that the ER is a tubular / vesicular organelle, right? Endoplasmic reticulum. It has like 2 types. The smooth and rough ER, smooth / rough portions of it. And we know that it's smooth if it doesn't have the ribosomes. So just remember that that is the difference between them. Okay? And what else? Let's see. Let me just move this real quick. And up there. Okay. So yeah, for the smooth ER and rough ER, let's write the difference. Let's draw the difference between them. Smooth and rough. Here. So a tape, that's a very straight line. It's fine. This is the smooth ER section, or SER. And then we have the rough ER. Rough ER, or RER. Alright? So for the smooth ER, you don't have no ribosomes. And this will be for steroid or liver cells. Just remember that. And for its functions, it would be for fatty acids and phospholipid making, plus calcium storage. Okay? Meanwhile, rough ER would be for, it would have the ribosomes, and let me see, just adjusting the mic. What, is it not working? Okay, there you go. This one's much better because I can't see the notes. Earlier I couldn't see it, now I can see it. So we have ribosomes there, and then for its, like, the membrane, this one's continuous membrane, continuous with the nuclear membrane. Okay, then this one would be for protein translocation. Okay, so that's the main difference. Smooth ER is more for fatty acids, while rough ER would be for disulfide bond formation, protein folding and assembly, and it would also be for signal peptide removal. Okay? And during mitosis, the smooth and the rough ER would disassemble into like tiny vesicles. And these vesicles would fuse back together after cell division to form your ER, to reform the ER. Okay? Now, let's go for the differential targeting of mRNA during translocation. Erase it all. Okay, so for the mRNA, we can start. Okay, so ribosomes, the burgers, they remain free in the cytosol or become membrane bound, but it depends on the ER signal sequence in the mRNA being translated. So, hello Astray, welcome to the stream. I think I think it's your first time popping in the live, right? Yeah, this one's we're talking about translation right now of from mRNA to proteins, but earlier we did the 1200 jumping jack celebration for the 12,000 subscribers, but I didn't do all 1200 all at once. We are at set 5. I divided them into 120 for each set, so 10 sets of that would be 1200, and I did set 5 earlier. So now we're halfway there. Whoop whoop. Oh, and going back to the mini lesson, we're talking about free ribosomes or them becoming membrane bound. So there, they could be the free ribosomes. Free ribosomes, no problem. Thank you for popping in. And you get your mini bouncy white bouncy fluffies also when you chat. So that one's nice. And if you type something like this, exclamation hat, it changes your hat. It changes your hat. So yeah, and see if I type this, I think one of those is my character, so it'll change its hat soon, really soon. There you go. See, it became the red hat. Yay! So yeah, the free ribosomes would, what's this called? Where is it? The proteins. Okay, free ribosomes translate the mRNA encoding cytosolic proteins, and they would form free polyribosomes that remain suspended in the cytosol. Water. Okay, and then these proteins are directly released into the cytoplasm. I think I need to draw the flowchart for this, because it's gonna be easier to see. Okay, so let me just draw it first. So we have the pool of ribosomes in the cytosol. At cytosol. Oh, no. This one needs a bigger picture. And a smaller marker. This one would work. So we have the common pool, right, of the burgers. Common pool of ribosomal subunits. Ribosomal subunits in the cytosol. And those would be like circles, maybe. And then you have the mRNA encoding a cytosolic protein. A cytosolic protein. Protein that remains free in the cytosol. So it's gonna float around, and that's the ribosome, right? And then this is the mRNA. And then it can make the proteins after that one. And then they're gonna be free polyribosomes. And they split up into the common subunits again. But note that the common subunits can also go towards, if they have an ER sequence, mRNAs that contain an ER sequence will direct ribosomes to the ER. So mRNA with signal sequence. Oh, let me change. I don't want my red cap. I want to change it into one of the fluffy emotes, because I also have the emote. No, not the book. Fine, I'll wait for it to finish, because there's like a 5-second wait after you change your hat. I'll change it again. There you go. Let's see if it changes it. But yeah, these proteins, the free ones from at the top, those could just be released to like the nucleus, mitochondria, or peroxisome. Meanwhile, these, the one that the mRNA that goes to the ER, those would be the ones that have the signal sequence goes to the ER lumen. And that's how it differentiates it. So just note that. But I totally forgot to say I'm present teacher. No problem. Thank you for coming by. I'm trying to learn as well. So that's why it's always helpful to have more pop in for the mini science streams. This one is just a review of like how like after the proteins get like translated or how they get there and where they go. So yeah. And this one is just note that there is the signal sequence. That's the main thing. Where's my... I have a mini note for it actually. A much better note, but I'll get that after discussing this part. Okay. Where's the picture? Okay. Oh, there you go. So this one is your ER. And you have the signal sequence. And that for the mRNA, and then they can just go back to that one. Poise subunit. Okay. Let me see. So we have the biosynthesis and maturation of insulin in this next page. Here, we can actually highlight the sequence of it from a proinsulin to proinsulin to just insulin. Okay? So, it starts with we have a signal sequence at the end terminus. Oh gosh, wait. Let me draw it. It's gonna be green. It starts with being green. No. Just focus on the signal sequence. I'll erase it. I'll focus on the signal sequence. So it looks like this. And then the signal sequence is over here. Okay? That's the signal sequence. I hope you don't get 1/4. Oh my gosh, get 1/4 sheet of paper. 1/4, mom. Yeah, that's what you always hear normally. Or get a crosswise, mom. Yeah. Yes, that's it's funny. Pro insulin. But now it's like iPads, right? iPads or laptops. Some students, but there are still some who write, so that's good. Proinsulin, that's pre-insulin, and then signal sequence at the end terminus. I'm gonna label it signal sequence. The signal sequence directs proinsulin, pre-PI, into the ER for proper folding to the ER. Okay? So now, once it's in the ER, once at ER, signal sequence is cleaved, and then you get proinsulin. So just this one. Proinsulin. After that, you will get mature insulin after you deliver it to the Golgi, because that's where Golgi cleaves C-peptide. So now you just have your insulin. Okay, so just as a mini review for how the insulin matures is it would go from pre-proinsulin to proinsulin to normal insulin. Pre-proinsulin would have a signal sequence that would direct it to being properly folded in the ER. I mean, proper folding and processing. And then once it's in the ER, endoplasmic reticulum, the signal sequence is then removed, And after that, the Golgi, it gets delivered to the Golgi, and then the Golgi will cleave the C peptide, and then you're left with just your finished insulin. Okay? That's good. That's good. Where's my sticky note? Sticky note, sticky note, sticky note. Next. Now, stepwise assembly of a transmembrane glycoprotein in the ER. Transmembrane, how to form glycoproteins in the ER. How to form transmembrane glycoprotein at the ER. Good question. So, first, we have the protein is inserted into the ER membrane. Protein is inserted into the ER membrane via it has an anchoring segment and all that, but essentially just put the protein is inserted into the ER membrane, and then after this one they fold. The extracellular domain folds inside the ER lumen. So protein is inserted into the ER membrane, and then the extracellular domain folds inside, and then the helices interact, and then you now have your fully assembled trimeric protein. Okay? Now, the next one is more important because it's co-translational translocation of the proteins into the ER lumen using your Sec61 translocon complex. This one, I really want to get this down. So again, this is co-translational translocation. Again, like you put it over there, of proteins into the ER to the ER using the Sec61 translocon. Very, very crucial. So it's there's like at least about 3 steps in a way. Okay, so number 1, you have your inactive protein translocator. Okay, because the ER segment right now, let's just draw them. This is the ER, this is the cytosol, and this is your membrane of the ER, and then you have your ER lumen, aka just the fluidy part, the inside of your ER. So here, oh no, I will need more space. No. Okay, fine. I'll just draw it over here. I'll draw it here. There's a line. This is your ER membrane, ER lumen, and then you have your cytosol. Okay, so you have your inactive protein translocator. It's going to be here. Inactive protein translocator. It's still closed. Okay, it's still closed. And a newly synthesized polypeptide with the N-terminal sequence is above it. It's floating. Okay? Oh, no. I'll draw it in a different color so it's easier to see. Let's draw it this way. You have a blue one, the normal protein, and then this is your signal sequence. Okay? Next, now you have an active translocon. The signal sequence binds to the Sec61 translocon. So let's see. It goes through here and then the red one here. Again, this is now active. Translocon is now active. The signal sequence binds to your Sec61 translocon, which will then open this channel, okay? And the growing polypeptide chain is now threaded
into the ER lumen as the translocation continues. So the way that looks like might be here in the middle. The red one is now here. And then your growing polypeptide chain, it's gonna be slowly growing over here. All right, so again, you have your signal sequence. This one is going from the outside, the cytosol, into the ER lumen, okay? This delivery of the mRNA. So this mRNA has the signal sequence, and then the signal sequence binds to the Sec61 translocon. Now, while that's binding, the translocation of the protein will continue. So that's why it's co-translational translocation of proteins. And now after this one is growing, your let's see. You then cleave it. Okay? We have a signal sequence peptidase, which I'm gonna draw it in yellow. This one would cleave the signal sequence peptidase. There you go. That would cleave your protein so that the remaining one enters further here. Now it's just floating in the ER lumen, while your signal sequence would then be released to the membrane. It's here. So that's generally how it works, how the Sec61 translocon works, okay? So again, one more time. You have, I will draw it again. So how the co-translational translocation works. It works this way. You have your ER membrane. This is the cytosol. This is the ER lumen. You have the Sec61 translocon over here. And then you have a floating protein with the N-terminal sequence. All right? Hello, Sheriff. Greetings. We are discussing how proteins are co-translationally translocated, and I have fancy math letters going on also. Oh, let me change my hat. One moment. Yeah, I want to change it. Because if you type exclamation hat, you change your hat. So yeah. Okay, now we're just reviewing how it works. So you have the translocon. You have a floating protein. It's still inactive. And then after that, this signal sequence would bind to the Sec61 translocon, and then the translation would just continue here down here in the ER membrane. Will down here. It's just gonna keep going. And then after that, after it continues growing, you have your signal peptidase. Yeah, the peptidase would cleave it. And so your signal sequence is left here in the membrane. And then your growing protein is now floating in the ER lumen. There you go. That's how it works. Now, there is a thing called N-linked glycosylation in the rough ER. And how does that work? Well, we will find out. Okay? So let's see. Here. Nope. There you go. So we have N-linked glycosylation in the rough ER. Rough ER. All right. Let's slowly but surely talk about this one, because this is gonna be fancy. I'm drawing the rough ER here. No, I'll make it more squigglier. The rough ER, it's gonna be like this. Rough ER. There you go. No, I'll write the words inside just so I don't get confused which one is which. Yeah, this is the rough ER. Now, you have your dolichal linked oligosaccharide assembly. Essentially, in the ER membrane, how can I say this? GLC3 man gluc NAC2 assembled on the dolichal phosphate in the ER membrane. Because this is essentially your dolichol with the phosphate, and your mannose and all that. A couple of mannoses. And, the nags. Phosphate, and nag. Okay, fine. I'll draw it. Your dolichol, your phosphate, your N-acetyl glucosamine, a couple of mannoses, and glucose. Okay, that's the sugar, and it looks like this: bam, 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 something like that. This one, you transfer. Who does the transferring, and why do we want to do it? Okay, we want to glycosylate them or put sugar molecules on your either growing or finished growing proteins so that they can do a lot of different things, because it depends on how glycosylated they are, if they can do their function. So right now we're talking about the OST, oligosaccharyl transferase. That's the one doing the transferring to one of the amino acids. Okay, this is an amino acid from a growing from an mRNA. Okay, that's it. This one's the mRNA. This one's your protein that's growing. And then one of them, for example, asparagine. Is it asparagine? ASN. Yeah. That we would put one of these there. Okay? To the ASNX SIR or threonine consensus sequence. So we trim it like glucosidases and mannosidases would remove glucose and mannose. And then we fold it like chaperones would assist in the folding. And then after that, the trimmed glycoprotein would move to your cis Golgi now. So again, after this one is finished and maybe glycosylated, they would proceed to the cis Golgi. So that's the N-linked glycosylation in the rough ER. Now, I will connect that with the thing earlier, aka the signals and all that, because this N-linked glycosylation is just a sneak peek. Okay, one more time, and I'm gonna bring back the Sec61 translocon that I talked about. So, again, this is co-translational. Translation, I cannot spell. This is co-translational translocation of proteins into the ER. And it is mediated, or a thing that helps it is the signal recognition particle pathway, or SRP. Okay? So now, you have your... There's a lot of steps, but I'll just do... I'll do 1 to 6. That's the number of steps. So for step 1, the signal sequence would... I'll just draw it instead of writing the words. This is your membrane, right? This is your membrane. ER lumen, ER membrane, and cytosol. So you have the signal sequence emerges from the ribosome and is recognized by the signal recognition particle. So where is that? The SRP. Wait, let's draw the burger first. Let's draw the ribosome. This is the ribosome. This is the mRNA. this mRNA, and you have a growing protein with a signal sequence. That's step one. Who will see it? The SRP will see it. Your signal recognition particle will recognize it, and translation will slow down. Okay? That's step 1 and 2. Signal recognition particle will identify it, and then it will slow down. After that, the SRP ribosome will bind to the SRP-ribosome complex will bind to the SRP receptor. Okay, what does that mean? So in here, on the membrane, you have this alpha and this beta subunit. This is the SRP receptor, signal recognition particle receptor, aka a protein on the membrane where the SRP and this ribosome binds to. And the reason why it's called SRP ribosome complex is because at that point your ribosome would look like this. You have that, the burger, and then your N-terminal, your full other protein. Oh, where's the blue one? Full other protein. And your SRP is no, it has to be blue. Your SRP is over here. Okay? So that's how it looks like. Your SRP receptor is where it docks. That's the docking protein. Your SRP and ribosome complex will dock on the docking protein. And this interaction is just stabilized by your 2 GTP molecules, one to the SRP and one to the alpha subunit. But that's just a mini detail. All right. Now, step 4, your SRP. Wait, 1 second before. Yeah. The nascent polypeptide chain is transferred to the Sec61 translocon, which opens the gate. What does that mean? 1 second. Erase. There you go. So, you have the Sec61 translocon. Remember it from earlier? Yep, that's how it looks like. Your nascent chain is transferred here. Again, how does it look like? It goes here and then it would grow. Slowly grow. The nascent chain is transferred to the Sec61 translocon. The gate opens. And the gate opens. And then your green thing, the SRP, and the SRP receptor would dislocate from the translocon. So it's not there anymore. The green and the receptor is not there anymore. Translation would then continue. And there you go. Translation continues. It's gonna continue growing. And GTP hydrolysis by the SRP receptor. Oh, that's just the part where you recycle the SRP. But the signal sequence after it continues growing, it's removed by, where is it? The signal peptidase. Detach it. Signal peptidase removes it, and soon it will be degraded. Translation continues. And you have your growing protein, right? And then this protein would have some carbohydrates added, your sugars, such as this one, at very specific amino acids, and the proteins are then folded by the chaperone, and translation ends, and the full protein is folded completely, and the translocon would close. So this very orderly process is very important because that would ensure that secretory uh, membrane proteins are properly synthesized, translocated, and modified before they go to the Golgi. All right. Yes. Hello, Endless Cash. Welcome to the stream. This is where we talk about the translocation of proteins. Oh, I have to introduce myself. I forgot. For the new folks. Hi, I'm Millennium, Isekai'd Mask Knight Empress Scientist, and I do Pal World Pokemon, science, and sometimes I do the science questions while walking on a treadmill, and sometimes jumping jacks as well. Because why not? You know, we only live once. Why not do jumping jacks? Right? Okay. So, again, going back to this one, we are in the ER lumen. I will draw it again. I want to make sure that I get this. I put this in my brain because this is an important uh, lesson. Let's see. Yes. That's what we do. Okay. So again, we have the ER lumen. Oh, by the way, since you typed in chat, you have, what's this called? You will have your mini character, and then you can change your hat. If you don't like your hat, you can change it using this, like that. And then after your hat changes, it's good. I think there's like a 5 seconds gap between each change. So yeah, that's how it works. All right, this is the ER lumen. Your ER membrane, and then you have your, where are we? That's the cytosol. All right. So we have the protein, the ribosome. Hi. So it's that one. You have your ribosome. This is your mRNA. This is the growing protein. And this one. And then your signal. Okay? You have a signal. Who sees it? Your signal receptor. What's the letter P? Signal recognition particle. Oh my gosh. The signal recognition particle would see this, and then they both bind to the alpha and beta subunit of the signal recognition particle receptor. Both of them will bind here. The way that burger looks like now is circle, circle, line, and then your protein. Oh, it has to be blue. Your protein, and then your signal sequence. Okay, what happens after that? The signal sequence will then go to the Sec61 translocon. Just the protein. It will go here. And then the gate opens, and that's why your protein will continue being translated, and then your SRP and all that jazz will stop being connected to the Sec61. Your protein will continue being translated here. Oops. It will continue being translated. Oh, wrong color. It will continue being translated, and at some point, the signal peptidase will cut this off, and your signal sequence will then be degraded. And then your fully finished protein could be modified, sometimes fully finished, or it could also be modified while it's still attached there. So the way it's modified would be on certain amino acids, some sugar molecules will be attached to it, and what's this called? The thing that the technique, not kagibunshin. The process would be called N-glycosylation. And the one who does that would be the OST, or oligosaccharyl transferase. And it would depend, I mentioned earlier, if the protein is already, the Naruto moment, it's gonna happen. For languages, I can do English, Filipino, some Kpop words, and anime. That's what the fruit salad is. So yeah. Oh, I mentioned earlier about if the protein could be already finished growing or if it's still growing before you attach your carbohydrates or the sugar. And that's because if it's OSTA, oligosaccharyl transferase A, it's gonna be translational. But if it's OSTB, it's gonna be post translational. All right. Now so that's it's slowly becoming more detailed. Oh, speaking of like anime, I watched. Okay, for it's gonna be the normal Naruto and then the Shippuden and then there's the Boruto part. I really like the pre Boruto, the normal Naruto and normal Shippuden. That one. And as for all the other animes, it would be Attack You Hitman Reborn. I think the description, I put it in the description too. All the different animes that I finished. Oh, the Bunny Girl Senpai, the Loop 7. I finished Kataki. Oh, I already mentioned all seasons of Kataki Hitman Reborn. And what else? Oh, why? 3D Kanojo. That one's really nice. Golden Time. Really nice. And what else? What else was it? Golden Time. Oh, there you go. Quintessential quintuplets. I hated the ending of that. Assassination Classroom. Haru Ride. The blue and pink movie. The To Every You I've like loved before and all that. Buddy, the Buddy Daddies, Charlotte, Chihaya Furu, Code Geass, Free, Fruits Basket, Fugo KG, Gakuen Babysitters, Horimiya, Howl, Hyoka, K-On, Love is War, World God Only Knows, Kimi Todoke, Kuroko no Basket, Kimi no Nawa Koroshi Ai, and other stuff. So lots of different things here and there. The My Anime list is going crazy. So again, that's also a part of my brain, aside from science, it's anime. So as translation begins, so this part would be the GTP dependent regulation of the signal sequence of the SRP pathway. Okay? So as translation begins, SRP would bind to the signal sequence, which pauses the translation. Okay? So again, SRP binds to the signal sequence and pauses the translation. And then your SRP GDP complex would bind to the GDP for GTP. Just remember the GTP would power it. One of the GTPs would be on the SRP and another one is just floating there. Okay, I've watched some anime on your list. Very nice. Anime is everywhere. That's just from A to K, I think. I didn't write the K until Z because the description would be too full. Oh, and manga as well. Yes. For Quintessential quintuplets, I really disliked the ending. I was like, why did that girl win? My chicken was supposed to be the winner, but the author had different plans. So yeah, for 2 models of protein translocation into the ER, we have, let's see, we have the signal hypothesis and membrane trigger hypothesis. Okay, so let's go 2 hypothesis. Oh, 2 models. Well, 2 models of protein translocation into the ER. So you have the signal hypothesis, which I kept on saying, signal hypothesis, and the membrane trigger hypothesis. All right. So the signal hypothesis would be there's a signal sequence on the newly synthesized protein which directs them to the ER. the one I said before. And this is the widely accepted mechanism for co-translational. But you also have the membrane trigger hypothesis, where instead of requiring like a dedicated protein pore, this one would suggest that the signal sequence would cause the proteins to fold and allow them to enter the ER directly. I'm confused. It sounds weirdly the same. So one moment. Instead of requiring a dedicated protein pore, this model suggests that the signal sequence causes the proteins to conform to allow them to enter. Oh, to fold. Okay, so I think signal hypothesis would be it's just they have the ID card and then they can enter the ER, but the membrane trigger would be the ID card would let them fold, and then folding would allow them to pass through the ER. Okay. Okay. So what is this? There's like an experiment that was used to study the co-translational translocation using an artificial mRNA system in a microsomal membrane preparation. Okay? So it looks like this. Let me draw it. One moment. Drawing. It looks very weird. This is the ribosome. This is your mRNA 5' mRNA, the 40S ribosome, 60S ribosome, 40S and 60S ribosome. And then you have this is your membrane. This is the Sec61 translocon. Okay? This is the Sec61 translocon. This protein, the different amino acids will be like this. And then the tRNA is somewhere. Oh, the ones that's like still growing because it's attached to this ribosome. Nascent protein just means it's the protein itself. All right. So an artificial mRNA is translated by a ribosome. The nascent protein would grow and passes through the Sec61 translocon. And then you have your cross-linking agent, a yellow agent. It's applied to direct the molecular interactions between the nascent protein and Sec61A. And that would confirm that the translocon facilitates protein insertion into the ER. What? Let me just read it. One moment. The nearsightedness is crazy. Cross-linking agent. Okay, the cross-linking agent is over here. Cross-linking agent. There you go. It's applied to detect direct molecular interactions between the protein and Sec61A, confirming that the translocon facilitates protein insertion into the ER. Oh, okay. Because it detects the interaction of it inside, then that's why it sees it. So it would support the signal hypothesis where, yes, it is about biology stuff. It's molecular and cellular biology stuff and some experiments. Sometimes I do biochemistry as well. But yeah, I just have to go through this one first. So yeah, good question. I'm trying to learn how to yap more about it. Yeah. Right, so what was it? This would confirm that Sec61 is the protein conducting channel. Yes, it is. And this experiment is in vitro, showing how the proteins enter the ER. Okay. Next. Next, post-translational. Yes. We learn a lot, hopefully. Hopefully I remember this one. Ah, I have to remember it. Stress. Next, we move to doing post-translational. Post-translational. Post-translational pathway. And this one is about the Sec 62. Oh no, another thing. So, co-translational is Sec 61, and it seems like Sec 62 is for post. Har. Alright, so I think I can just talk about this one. Co-translational is when your protein is translocated. Oh, table time. Table. Wow, very straight line. Thank you very much. So this is co-translational, translational, and post-translational. Translational. Okay? So co-translational is your protein is translocated or moved to the ER, translocated to the ER via who? The Sec61 translocon. But your post-translational would be it's Sec62 dependent, where it's used for proteins with less hydrophobic signal sequences. Okay, so co-translational, you really want your SRP, but why is it sometimes the post-translational version? It's for proteins with less hydrophobic signal sequence. Hydrophobic. Okay. The protein is fully synthesized in the cytosol before being targeted to the ER. It's more common in yeast and mammalian and for small proteins because they're released from the ribosome too quickly to even engage or interact with SRP. Okay. So co-translational might be for the big boys where the SRP is needed. Post-translational is for simpler stuff and the ones that are quickly made. Okay. Okay, we finished that one. We have one moment. There's a coupling. There's a coupling between translation and translocation, right? Co and post. So there's the chaperone movement, such as the heat shock proteins, which help maintain the unfolded or translocon competent conformations. So, and they prevent premature folding or actively unfolded proteins. So that is the heat shock proteins. Heat shock proteins. They prevent they help maintain the unfolded or translocation competent conformations. Either maintain the unfolding or maintain the folding. Good to know. Okay. Now I will let me see if I can draw it, because this is talking about chaperone assisted folding proteins. Chaperone. Let's see. We have chaperone assisted folding proteins. Protein folding. What? Words can be switched, but not here. Protein folding and translocation. Specifically in the mitochondria and in the ER. So proteins that are synthesized in the cytosol are assisted by HSP70 and all that to prevent premature folding. Mitochondrial translocation is very post-translational. ER translocation is co or post, which will ensure that the proteins are folded correctly in the ER. And misfolded, what happens to the misfolded? Misfolded proteins are either refolded, disassembled, disassembled, or degraded via lysosomes, particularly under heat shock or cellular stress. Now, we have, I don't know the stuff that you're yapping about, but I'll still listen in case something tickles my curiosity or something. Thank you. Yes. The next, I think after this one would be Golgi apparatus, so hopefully we'll get there. But yeah, I'll just keep yapping. Thank you very much for listening. The note SRP pathway is not the only pathway, and just the general concepts of protein targeting into the ER. Okay, ER summary. Into the ER. Let's see. Into the ER. You have the signal sequence. Signal sequence. The machinery is your signal recognition particle. Recognition particle. You have the Sec61 or Sec62, and then energy would be ATP and GTP. Now, moving on to the mitochondria. Mitochondria time. What is the mitochondria? Mitochondria is the powerhouse of the cell. What is CRISPR? Clustered, regularly interspaced, short palindromic repeats. Again, CRISPR is clustered, regularly interspaced, short palindromic repeats. Do nice? Yeah. But yeah, for the mitochondria, how do you target proteins to there? Good question. So the mitochondria looks like a bowl, right? With some squiggles like that. So this one has 4 components. You have the outer membrane, the inner membrane space, aka here, inner membrane space. And then you have the inner membrane. And then you have the matrix. And that's where oxidative phosphorylation and synthesis of metabolites happen, including amino acids and all the other stuff. This one, you have the Tom complex here, okay? The Tom complex would be your general entry point for all pathways. And during and after synthesis, your HSP, the heat shock proteins, would try and bind to maintain them. But yeah, there are 5. Oh my gosh, there are 5 major protein import pathways to the of the mitochondria. Why are there 5? Just one, please. Okay, so we have the Tom 40. It's very crucial. My note says asterisk crucial. But why is it crucial? So you have the pre-sequence carrying pre-proteins. It's folded by the translocase. Oh, I want a picture. Picture with steps. Picture with steps. Where is it? I like this one. You have... Okay, I can draw this part. Picture with steps. Alright. This one, we are now in the mitochondria. Don't get it mixed up. That's the outer membrane. Outer membrane. The inner membrane space. Inter membrane space. Why is it called intermembrane space? It's space between the membranes. Oh, wow. Okay, this is the inner membrane. Inner membrane now. And then You have the matrix. Oh, by the way, outside is the cytosol. Cytosol. Okay, nice. So you have the Tom. This is the Tom. Tom, T-O-M. Proteins designated for the mitochondria have a pre-sequence. So again, they have a signal that says, oh, we gotta go here. This is the signal sequence of the mitochondria. For the mitochondria. So the Tom, translocase of the outer membrane. That is funny. Translocase of the outer membrane is over there. It recognizes these proteins. The Tom 20, there's a couple of Toms that would act as receptors. But you have the Tom 40, which forms a channel that allows the proteins to pass from the outer membrane into the inner membrane space. So you have the Tom 20 and 22, which recognizes them, but the Tom 40 would form the opening where your protein can go here now to the inner membrane space. And then intermembrane space. And then you have your Tim. Guess what the Tim is for? It's another receptor. I think this one's translocase of the inner membrane. You have the Tim. It captures the protein and hands it over to another Tim 50, and hands it over to Tim 23, which forms a channel with Tim 17. Wow, it's a lot of Tims. So if we want to try and remember, let's see. Tom, I'm gonna close my eyes so I can see it in my head. Tom 22 and 20 would see the incoming protein with the protein signal for the mitochondria, and it says welcome home. And then the protein goes through there, and then Tom 40 would have the gate open, and then it passes it to Tim. Which Tim? To Tim 50, and then gives it to Tim 23, which forms a channel to Tim 17. Oh gosh. So 20, 22, Tom 40, and then Tim 50, and then hands it over to Tim 23, and then talks to Tim 17. Tom, Tim. Okay. And then the membrane potential helps pull the positively charged pre-sequences through Tim 23. Oh, another one. Tim 23 into the matrix. So lots of things going on. But essentially, it slowly passes through a couple of Toms and a couple of Tims until it reaches the matrix. Now, let's see. Again, chaperones help them fold properly, or else they are degraded. Okay. Tom, and Tim. Good. Finished. Now, let's talk about the nucleus. So, we did the mitochondria. Oh, we did the ER. It was the Sec61 and Sec62. And then we did the mitochondria, which was about the Toms and the Tims. And then we now have the nucleus. So what's up here? What is going on over here in the nucleus? I don't know. Does biology not have calculation, mostly memorizing our names? Good question. It also has the numbers, especially in bioinformatics. And then you may have heard of things like the Punnett square, and stuff like that. Plus, there's the RNA-seq. The sequencing has a lot of calculation stuff. RNA-seq would be sequencing your RNA, like a different type of genetic material. And then another one, there is a technique called polymerase chain reaction, the PCR, which would, if you have the DNA, you just want to times 2 times 2 times 2 it, but there's a lot of things going on there as well that might, like the temperatures and all of that. There is also a calculation here, but a lot of it is concepts that you have to ingrain in your soul until you dream about it and then wake up and you're like, oh my gosh, what was that certain receptor again? Who was it? So that is the headspace. But yeah, yes. And then now we are in, oh, the nucleus. So, what is the nucleus? The nucleus is where, the nucleus is where your genetic material is housed. It is the house of your DNA. And your nuclear envelope, aka, if this is the nucleus, your nuclear envelope is a double membrane. Nuclear envelope is a double membrane whose lumen is very continuous of that with the ER. And the surface of the nucleus is studded with nuclear pores. With nuclear pores, don't forget that. And small proteins can just diffuse across it, while large proteins would need the nuclear localization signal. So another signal, nuclear localization signal. And it consists of different monopartite, bipartite, and all that. But the NLS, the nuclear localization signal, can be located anywhere in the protein, and it's not cleaved after transport into the nucleus. Okay? So, and there's also the nuclear pore complex. I'm going to talk about that in a bit, but that's an important thing. And yeah. Now, the nuclear pore structure, all proteins in the nucleus comes from the cytosol. All right? Oh, I was going to move this like mini camera. But yeah, all proteins in the nucleus comes from the cytosol. Again, all proteins in the nucleus comes from the cytosol. Proteins are folded and diffusion cutoff is about 40 kDa. The steps for active transport, it's very post-translational translocation. The NLS, the nuclear localization signal protein. Oh no, I need a picture. It would bind into the importin A via the nuclear localization signal. You have RAN GTP. I don't have a picture right now. But those are the important terms for it. Where is it? Oh, this is just the RAN. Okay, there you go. There's a picture. Don't worry. Thou shall not panic. We have a picture. 1 second. I just need to make sure that this is the right picture. Okay, there you go. It is. All right. We now have importin alpha transport cycle. So this one, your NLS containing the protein which have the NLS protein binds to importin A, binds to importin A. And importin A has the NLS binding site. That's why it can bind to it. So again, your protein, so that it goes to the cytosol, to the nucleus, it would have, what's this called? It would need your NLS, the nuclear localization signal. And once it has that, the NLS would be able to bind to the importin alpha because it has the binding site for it. Importin A, importin alpha binds to importin beta. Interacts with importin beta. And now you form a trimeric complex of your protein plus important alpha and important beta. After that, the complex would bind to a thing called the NPC. It's not non-playable character. It's the nuclear pore complex. And the complex is moved to the nuclear side where it has very, very high RAN GTP. Wow, that we are just at step 3 out of 10. Calm down. Calm down. Okay. So again, let's not forget steps 1 and 2. The NLS, the protein which has the NLS would bind to important A because important A has the binding site for the NLS. Important A talks to important B or alpha, and now you have the trimeric complex, which then talks to the NPC, nuclear pore complex. Nuclear pore complex translocates to the inside part of the nucleus because the nuclear site, because that one has a high RAN GTP. Now the next question is, what's up with RAN GTP? So RAN GTP binds to important B. We know that, which terminates the translocation and disassembles the important hetero. Ooh, I need to draw these like they are friends. Yeah, non-player character. So let's see. This will just help us visualize it. Your protein, which has the NLS. Let's write it in gray. Your protein, which has, what is that? Okay, your protein, which has the NLS would bind to important A, would bind to important A, important alpha. Then this one talks to important beta. Okay, good. We have that. Now, this whole thing will talk to the NPC, the nuclear pore complex. So now we have the protein and 2 things. And now your NPC will bind to important B or alpha, terminates translocation, and disassembles these. Okay. The NLS protein is released from important A alpha. Oh, because now it's inside the cytosol. The nucleus. It has been delivered. That's why. Okay, now all good to go. Yay. Happy. RAN GTPAs. Oh, you have the RAN, H plus P70. Note, the NLS, they're not removed after transport. So that's the main difference of the signals. Okay. So we discussed the ER, mitochondria, and the nucleus just as a mini review. ER, well, erase. Trying to deliver proteins is hard work, very hard work. So this is your protein. If we want it to go to these 3 at least, ER, mitochondria, and then the nucleus. ER, what was it? This is the Sec61, Sec62. 61 would be where the SRP is needed. Mitochondria that's the TOMs and the TIMs. And then nucleus is where nucleus would be for NLS and NPC. And important alpha and beta. Beautiful. Wow, I can't believe it's been like so long since we summarized this one, because it's been about 1 hour and 30. That's why I wanted to discuss this properly. Yeah. ER and mitochondria. Okay, this one's finished. I'm just sticky noting. Sticky notes. The sticky notable section. Whoa. Sticky note. Okay. Experimental question. So, if we are studying... Oh, I like this. I have to go through this one. If we are studying a newly identified mitochondrial... Oh, I have to move. 1 second. I didn't notice that my eyes kept closing because the camera wasn't capturing the eyes. 1 moment. Oh no, I can't move it. I'll move it later. So we have... Which one was it? Yeah, we're studying a newly identified mitochondrial matrix protein. Alright? We're studying... Oh, I cannot see it. We're studying a newly identified matrix protein. And we think it might be dependent on the TIM23 translocase of the inner membrane. Alright? To confirm this, we treat the cells with a drug that inhibits TIM23 function. Alright, okay. How can we use Western blotting to determine if protein X is mislocalized due to TIM23 inhibition? So we know that Western blotting is a technique used for seeing if a protein is there or not. And it would be the SDS page. Okay, let me draw it. I want to draw it. It's a mini review. Because it would just help me visualize how it all works. Let me see. So the way Western blot works is you have your SDS page, aka you have a protein, you coat it with sodium the decyl sulfate just to denature and stuff like that, or just so that when you do your gel they would only transfer because of their size and not because of their charge. All right. Oh, thank you for reminding me about the hat. I want to change my hat also. There is a dab hat. I want the dab hat. I hope I get the dab hat. There's a dab, there's a sparkly, there's an angry hat, there's a sad hat, there's a very happy hat, there's a sparkly hat. There's a lot of hats. There's more than 25 or 50, I think. So yeah, you can go crazy with the hats. So yeah, and then when you do their page, you transfer, and now they look like this. And then you transfer it to... Okay, so here's the thing. Let's say your protein is around this size, right? But you can't guarantee that there's only one protein. Because what if you got all the proteins? What if this is 5 proteins stacked on top of each other? So that's why you transfer them first to a membrane where now they're flat like that. And then you put antibodies, okay? You block it first, and then you put antibody 1. Let's say this is your protein. But you also have other proteins there, right? Let's say. But with your antibody, the first one, with your first antibody, it's only going to bind to the protein that you want. So that's good. And then you wash it, and then you have your antibody number 2. Antibody number 2. Ding ding. And this is the one that fluoresces. And that's the one that you'll see on your chemifluorescent result. So that's how you can see oh, I see it. It's really there. The protein that you want. So how can we use Western blot to see if our dear protein is related to TIM23? All right. If the protein is dependent on the TIM23 complex. So we have to compare the protein X levels. Let's say in the mitochondria versus the cytosol versus the cytosol and intermembrane space in treated versus untreated cells. I have to write that properly. I can't see what it means. Okay, 1 second. We have this. All right. So at least in control cells, protein X should be in the mitochondrial matrix. But in the TIM23 inhibited cells, the protein X should be in the cytosol and intermembrane space. Okay, we have control and treated, aka with the drug that stops TIM. Okay. So what are our expected results? So in the control, normal TIM function. That means we would be able, this is your mitochondria. If your TIM is okay, this is Tom, that means the protein can freely pass and then you would see it in the matrix. Yes. But if it has the drug, that means it is stuck in the cytosol and the intermembrane space. Membrane space. So that's what we're comparing. We're comparing the protein levels in the mitochondrial fractions, like the cytosol versus intermembrane space versus the matrix. And how do we do that? Well, we have the target protein. We would be able to see, let's say this is the blot. This is your final thing. We have the target protein. I don't know. We have the target protein. We need 3 markers. A marker for cytosol, aka a protein in the cytosol. A protein for an intermembrane space protein. And a matrix protein. And in the areas where, let's say, if we see the matrix protein and target protein together, that means that's where the target protein went. And in the treated ones, I don't think we'll see this combination. Oh yeah! Okay, how can we use flow cytometry to quantify mitochondrial integrity following the inhibition? So we have flow. What is flow cytometry? That one is a cell, 1 by 1 cell, right? Each one. So since TIM23 relies on the membrane potential, if you stop TIM23 with a drug, it might disrupt the membrane potential, which would affect the whole mitochondrial integrity. So just compare the mass of the mitochondria. And if TIM23 inhibition affects your potential, aka a very lower fluorescence intensity, then that means you didn't observe it. Okay, essentially, Western blot would confirm where your protein X is once you inhibit the TIM23. Flow cytometry would just show the overall mitochondrial health depending on the membrane. So now we have proteins A, B, and C. ER and ER protein, mitochondrial protein, and nuclear protein. Design an experiment to determine which signal sequence each protein contains. No, okay. And clone each protein's... Okay, first, let's go with A. Design an experiment to determine which signal sequence each protein contains. We clone the protein's DNA into a plasmid. What? Let me just draw it again. What is it asking? What is it asking? Here. Alright. So, design an experiment to determine which signal sequence each protein contains. All right. So that means we want to see this signal sequence from the ER one, from the protein one, and from, no, that's not protein. From the mitochondria, and then this one is ER. Come on, where is my mark? This is the ER. No, nucleus. Okay, let's see. We will clone each protein's cDNA into a plasmid. Clone, okay, this is a protein. Okay, good. Clone the protein's cDNA into a plasmid. So that means finding its specific gene, put it into a plasmid with the fluorescent tag. Okay, the tag being like that. Put that into a mammalian cell and use fluorescence. Okay, and to confirm, the sun is rising. Yes, in time zones, the sun might be setting, rising, or it's the middle of the day, or it's the reverse of the middle of the day, aka middle of the night. Yes, but don't worry. If the lights, the lights can be turned on or off. If the sun is rising, that could be your new light bulb. It's a really big light bulb, though. Mammalian cell. Oh, where is it? Oh yeah, we clone it, and then we would check the fluorescence of it. Okay. The where it is. It's okay. Now predict the outcome if protein A's signal sequence were removed. That means it's not gonna be delivered into the ER. So it can't be, yeah. If the signal sequence is removed, then it won't be seen by the SRP. If protein C's nuclear localization signal was swapped with protein B's mitochondrial targeting signal, then that means you would see protein B in the nucleus and you would see protein C in the mitochondria because you swapped them. Now, a scientist mutates the arginine residue in the presence of a mitochondria. A scientist mutates the arginine residues in the pre-sequence of a mitochondrial matrix protein to aspartate. How would this mutation affect mitochondrial import? So arginine, that is a positive one. We need it so Tom and Tim can see that protein. But if it becomes aspartate, a negative residue, then that disrupts the import. All right? So if we want to rescue it, we just need to put in a genetic mutation or reintroduce a gene that will overexpress some heat shock proteins to help with its folding. Good. Oh, there's another page. Okay, you inhibit GTP hydrolysis in a cell and analyze protein localization. Predict which organelle's import would be disrupted, ER, mitochondria, or nucleus. I think nucleus. Nucleus would be most disrupted because it needs RAN GTP hydrolysis for the release. ER and mitochondria is less affected because it relies primarily on ATP. Now, if ATP hydrolysis is blocked, mitochondria is most affected because ATP-dependent heat shock is essential. Okay. So again, if GTP is blocked, who is most affected? Let's write it here. If GTP is blocked, nucleus is greatly affected. But if ATP is blocked, mitochondria tom is greatly affected. A cell line with a mutated heat shock protein 70 is analyzed. What defect would you expect if, where is it? in the mitochondrial protein translocation. So heat shock protein 70 mutations would stop it from being pulled into the matrix. So and that means they would just accumulate in the intermembrane space maybe. Co and post. You're studying a newly discovered protein that enters the ER. Describe an experiment to test whether it is co or post translational. Ay. Okay. We could do pulse chase actually, because pulse chase we would put in, let's say here, I'm gonna draw it. Let's see. Erase it. We can do it. Hasuiso. Jayo. Fighting. Ganbatte. Okay. Here. Where was it? To study if it's co or post. Pulse chase. I said pulse chase. We can do pulse chase because in pulse, you put in radio labeled amino acids. And so if those radio labeled amino acids are immediately put in the protein, that means it's co-translational, aka it happens immediately. But if it does not get integrated immediately, then that might imply post. Okay? Later on. You treat cells with a drug that inhibits TIM. What type of proteins would accumulate in the intermembrane space instead of the matrix? Oh, they would fail in the accumulator. How could you confirm the mislocalization using biochemical methods? Oh, we would just use Western blot, not Webtoon. Use Western blot. A mutation in RANGEF prevents our GTP loading on RAN. What's the immediate sequence for nuclear protein import? It cannot be imported if that's screwed up. Okay. How would the mutation affect nuclear export? It's greatly affected and blocked. Yay! So we finished the protein translocation, especially in the nuclear. What was it? What's the first one? ER, mitochondria, and nucleus. Whoop whoop. Nice. We can move on to the Golgi apparatus actually, but I think I may need to the rest of the voice for now. But thank you very much for watching and learning. Bye.